You're standing in ankle-deep surf, and the broken edge of a shell catches you across the top of your foot. It's nothing. A thin red line, gone in a few seconds under the waves. But your cousin, 4 feet away, goes pale. "Get out of the water," she says. "Sharks can smell 1 drop of blood from a mile away." You laugh, but you also start walking toward the sand a little faster than you meant to. That line has been repeated so many times, it barely sounds like a claim anymore. It sounds like a fact. Thank a movie about a rubber shark named Bruce for that one. But it isn't a fact. A shark could be a mile away, on the other side of a sandbar, past the buoys, and it would have no idea you exist. This is the story of what a shark's nose can actually do, which turns out to be stranger and honestly more impressive once you drop the mile and look at what's really going on underneath it. Start with what people picture. A shark out in the blue picks up a single molecule of blood and draws a straight line to you, like a compass needle finding north. That's not what's happening, and here's the first correction worth making. A shark doesn't receive a location. It doesn't even receive a ready-made direction. All it gets at first is a signal arriving at its nose. Figuring out where that signal came from is a separate job entirely, and the way sharks solve it is stranger than the myth ever gave them credit for. Start with the hardware. A shark's nostrils aren't for breathing. Sharks breathe through their gills. The 2 openings under its snout, called nares, exist purely to sample water flowing past. Water runs in through a groove, over a set of folded tissue called the olfactory rosette. Picture the pleats of a coffee filter, packed with thousands of sensory cells, and back out again. Nothing crosses into the throat. It's a one-way scent chamber, and a shark has 2 of them, one on each side of its head, which turns out to matter enormously. For decades, the working theory was that a shark tracks a scent the way you'd walk toward a stronger and stronger smell of smoke, comparing which nostril smells more concentrated, and turning toward the stronger one. Then, researchers strapped a kind of scent headset onto small dogfish sharks, feeding controlled puffs of odor into each nostril separately, with total control over timing and strength. What they found upended the old theory completely. The sharks weren't comparing strength at all. They were comparing timing. Whichever nostril caught the scent first, even by less than half a second, even when that nostril's puff was the weaker one, the shark turned that way, every time. Here's why that distinction actually matters. An odor underwater doesn't look like a smooth, expanding cloud. Picture it instead as a shredded, tumbling ribbon, thick in patches, empty in between, twisted apart by turbulence the way smoke breaks apart in wind, rather than drifting as a neat cone. A shark crossing that ribbon at an angle doesn't get a clean concentration reading. It gets a strobe. Scent, nothing. Scent, nothing. Comparing loudness between 2 nostrils in that kind of chaos is nearly useless. Comparing which nostril heard it first, though, is a signal a shark can actually trust, patchy plume or not. Timing beats strength, because in real, moving water, strength lies. It's a strange thing to realize an animal is measuring stopwatch splits between 2 nostrils a few centimeters apart, using a brain the size of a walnut. Nobody expected that. For most of the 20th century, textbooks confidently described concentration following as settled biology, the kind of detail nobody bothered to double-check because it seemed obvious. Sometimes the obvious explanation is just the one nobody got around to testing properly yet. So how sensitive is the equipment doing that job? Laboratory measurements have placed shark detection thresholds for certain amino acids, the kind of compound a wounded animal releases, down in the nanomolar range, an extraordinarily faint concentration. Popular science writing likes to translate that into something more vivid, a drop of blood in an Olympic swimming pool. That image is worth keeping, with one honest caveat attached. It's a rough illustration of a lab number measured with pure amino acids, not a literal blood in a pool experiment. Either way, the underlying point holds. The receptors themselves are remarkably fine-tuned instruments. But here's the catch, and it's the whole myth quietly falling apart. A sensitivity threshold only tells you how faint a signal can be once it reaches the nose. It says nothing about how far that signal had to travel to get there. And in the open ocean, there is no single, universal detection distance. A large carcass leaking scent continuously into a steady current can build a plume that stretches for hundreds of yards. A single brief drop, dispersing once into open water with no current doing it any favors, may never form a trackable plume at all. The range isn't a fixed radius built into the shark's head. It's created jointly by the source and the water, and it changes every time either one does. So how far does one actual person, with one actual small cut, realistically project a scent? An informal demonstration filmed off the Bahamas tried to push the question about as far as it goes. A content creator and a marine biologist anchored well offshore, in water known to hold reef sharks and blacktips, and released real human blood at a rate of roughly 15 drops a minute, far more than a scraped foot would ever produce. No sharks approached to investigate. It wasn't a controlled, peer-reviewed range experiment, and the people who ran it said as much themselves. It can't tell you what every shark in every current would do, but it illustrates the point well. A minor human wound does not automatically generate a plume worth following. This timing mechanism has also inspired one of the leading explanations for the hammerhead's bizarre, flattened head. Spread two nostrils farther apart, and the timing gap between them, the moment the shark crosses a scent ribbon, gets larger and easier to resolve. That's a real, proposed olfactory advantage, but it's almost certainly not the whole story. A hammerhead's wide head also reshapes its vision, its electrical sense, and the way water flows around it while swimming, and researchers studying the shape tend to stop short of crediting smell alone. Evolution rarely settles for one reason when a structure does 5 jobs at once. Interestingly, sharks haven't been steadily evolving toward wider and wider heads either. The fossil record suggests the opposite, a slow narrowing back down across many lineages, which makes the hammerhead's shape look less like an endpoint and more like one experiment among many that happened to stick. It's also worth killing the other half of the myth, the idea that sharks are chasing the smell of people specifically. Sharks have often been billed as swimming noses, with a sense of smell that dwarfs everything else in the ocean. When researchers actually compared shark olfactory thresholds against ordinary bony fish, the numbers landed in roughly the same range. Sharks aren't operating on some separate scale of smell, and a larger olfactory organ doesn't reliably predict a lower detection threshold either. What they do have is receptors tuned, over hundreds of millions of years, to the amino acid profile of injured fish, seals, and other marine prey, not to humans. Human blood may register as chemically detectable, but detection isn't recognition, recognition isn't attraction, and attraction isn't an attack. 3 separate steps, and the myth skips straight from the first one to the last. Even where people and sharks genuinely share the same water, surf breaks, reef flats, popular beaches, unprovoked bites stay remarkably rare set against the sheer number of people swimming, surfing, and wading through it every single day. That's not because sharks are toothless. It's because a stray trace of blood almost never assembles into the kind of detectable, followable signal the myth assumes it does. And smell was never running the show alone anyway. It's one input among several, not the first domino in a fixed chain. A shark can be combining a faint scent trail with the low-frequency thrashing its lateral line picks up from something struggling, with what its eyes can make out at closer range, and, inside the last few feet, with the faint electrical field of a heartbeat, sensed through pores around its snout called the ampullae of Lorenzini. Different species and different situations weight those signals differently, and more than one is often running at once. Smell rarely closes the whole distance by itself. So the honest version of the myth isn't sharks can't smell blood. They genuinely can, at concentrations far fainter than almost anything else swimming in that water would ever notice. The honest version is that sensing something and finding it, across open current, from a mile, are 2 entirely different problems, and only the first one is something a shark's nose was actually built to solve on its own. Back at the beach, the cut on your foot is already closing over, rinsed clean by the next wave. Somewhere past the sandbar, nothing has turned to look for you. Not because sharks are gentle, and not because their noses are weak. They carry some of the most finely calibrated chemical sensors in the ocean. It's because the specific danger everyone grew up fearing was never really about the shark's nose at all. It was about a distance nothing in that water was ever built to cross alone.